I would like to welcome everyone back at this session. This is not a presentation session, but rather a very interactive session with questions to the different speakers. I will shortly introduce them. Then we will go. Then we will pose a question to each of them, and at the end there is ample room and time for questions from the audience for all the different speakers. All the people in this session had a different role in the Preventomics project, so they actually represent the different angles of the project from a biological sense, and I think they pretty well cover the whole project. I would like to start with saying that Claudia Favari is unable to come because she is ill. She has a fantastic replacement in the form of Pedro Mena, who is a colleague. Pedro Mena is a recipient of the prestigious ERC grant, and he works as an associate professor at the Department of Nutrition at the University of Parma. I will introduce all the people, but I will go through the whole list of people that we have here first. The second person is Radu Ghemis. He is an R&D researcher from the Microbiome Engineering Group at the LEITAT Technology Center. And he investigates the relationship between the microbiota, so the microbes in the gut, and the occurrence of different pathologies, including obesity. The next speaker is Professor Francisca Serra. She is a full professor in nutrition at the University of the Balearic Islands in Spain, and she is also research director, R&D director of the company Alimentómica. In both positions, she investigates the relation between genes and nutrients and how they affect obesity. Then we have Nuria Canela in the panel. She is director of the Omics science unit at Eurecat. So Nuria obtained a doctorate from the University of Barcelona, and then she moved into different omics technologies at in different places. And she really centers around both technological development, but especially also applying a number of different methodologies to study different biological systems. And finally, we have Josep Maria Del Bas in the panel. He was introduced before. He is the director of the Nutrition and Health unit of Eurecat, and also the scientific director of the Preventomics project. And he has a PhD in nutrition and metabolism from the Rovira i Virgili University at Tarragona, and spent quite some time abroad in Baylor College in the US and also in Milan in Italy. Okay, I will now go to the first person actually on the list, and this is Pedro Mena. And I would like to ask him why is it important to use food intake biomarkers in personalized nutrition? So why not rely on questionnaires?
Yeah. First of all, hello everybody. It's a pity for me to be here somehow, because I wanted Claudia Favari to be presented today. We wanted her, but unfortunately she cannot attend. And I said it's a pity because she's the person that is more able to answer all these questions. But since I have to reply, Jaap, I will try to do my best. I mean, why should we rely on biomarkers of food intake? Because to date, there is a need for more accurate measure when it comes to dietary assessment. All the tools that are available to date rely on consumers, on participants, rely on their time they have to reply to the question we are posing to them. But in order to have a proper coverage of the diet of a person, we need, let's say, about 1 hour with a person if we want to have a look at their habitual diet, or we are asking a series of efforts in time of times, in time of thinking about what they are relating to consumers if we ask for the foods they have eaten in the last days. And all the tools are key, are validated, are essential in the nutrition field. They have been using for many years, and I think they will be still the gold standard in the field. But they are relying on the consumer, on the volunteer, and not on objective measure, as for instance, a metabolomics platform can provide us. So in this case, what biomarkers of intake can allow is to have a complete fingerprinting of the diet of a person, relying just on an analytical tool that at the end of the day is an instrument analyzing a urine sample that you can collect in the morning, and consequently have a series of information on the diet of the person without much effort from the participant side.
Okay, but so I also understand. I'm also getting older, so you also forget what you have been eating, especially if it was quite some time ago.
Yeah.
But one of the problems with the questionnaires is that it is very difficult. It is easy to think, okay, what have I eaten yesterday or today? Maybe yesterday, but I can't remember what I've been eating 10 years ago. So do you think that if you start to measure things, that also the time resolution will improve?
Yeah. I mean, I'm also unable to remember what I ate yesterday even, no? So because of that, the importance of this tool, because they can cover what the mind cannot cover. In particular, as you mentioned, I think you are not old enough, Jaap, but think about a 70, 80 years old person, no? And perhaps you want to improve their dietary habits. They are one of the main target population in the nutrition field, but perhaps they are not able to remember what they have eaten in the last days, no? So because of that, the importance of this tool that at the end of the day rely on objective measure, and you don't have to take care about the memory of the person. And at the same time, I think it's impossible to cover what a person has been eating in the last year, for instance, through an analysis of a single spot urine. But perhaps having different spot urine along, let's say, a series of weeks can provide a very good overview of the diet of a person without, as I said before, almost any efforts from the participant side, but just from the analytical team behind the analysis of the sample. So at the end of the day, you don't have to rely on your memory. You don't have to feel guilty if you forgot something, but you only have to provide a few samples of urine.
Okay. Pedro, thank you very much for introducing yourself by answering this this question, and I will now go to Francisca Francisca Serra. Sorry, I have too many screens open. I'm sorry. So you are working, looking at the genes and gene nutrient interaction, and we are also measuring a lot of different parameters in circulation, in urine and in blood and possibly also in saliva. They all give an idea of what is going on. So can you explain us what you think the contribution of genetics will be to this field? And do you think there will be any contribution? Let maybe start with that first.
Well, good morning to everybody, and thank you very much for your presentation of myself. Concerning your question, of course, I think that genetics will contribute. Professor Ordovas already gave some message about this aspect. So in our case, in Preventomics, what we have done is to select a number of variants that can be related to the tendency, to obesity, to diabetes, to the inflammation, to those aspects that are under study in Preventomics. And also we have studied specific genetic variants that could be modulated by nutrition, by nutrients. So in this way, we think that we are able to give a specific recommendations to the persons, saying which is the best nutrient for to counteract the obesity tendency, or for example, in the case of the graph that Professor Ordovat show about if low carb or low fat diet, which is the best to you. So we are able to analyze this and to give the genetic recommendation on this aspect. So in this way, I think the genetics is another piece of information that goes together with the biomarkers, with the clinical biomarkers and the dietary biomarkers and so on. And in this sense can contribute to higher efficiency in the treatments for this person that has already a problem on obesity.
Okay. So if I understand well, that you are saying that, so if you measure things and you measure the metabolome and then you get a kind of status out of it, but if you want a recommendation, then by including the genetics, you more or less monitor the sensitivity of a person for a certain diet and how this change will affect it. Is that correct, or?
More or less. As Professor Ordovat said, we don't know everything. So we are based on scientific evidence that has been published, and we are using this data. So we are close to an estimation of what is going on, but we are not certain sure that this will work as should be.
Yeah. Can you give us maybe one example of maybe a gene variant that may impact which may impact the way you give dietary advice?
Oh yes, for example, there is the FTO, the FTO gene is a gene that predisposed to obesity in a very important way. And this can be modulated, for example, by exercise and also by age. When you are getting older, the influence of the gene is lower. But if you are exercising, this also minimize this interaction. If you are eating French fries, so you get a bigger increase in body weight if you have this genetic influence. So there is some sort of recommendation that can be given according to the genetic background.
Okay.
And this is only one of the genes that have been studied. We are analyzing 180.
Yeah. So I think this is very illuminating and very clear example. Thank you, Francisca.
Thank you. You're welcome.
I will now change to Radu. So Radu Ghemis works on the microbiome, and maybe you can first start to explain what the microbiome exactly is.
Well, thank you first of all, Jaap, for your great introduction. Well, the microbiome is the conjunction of all the bacteria that live with us and which give us a lot of different functions that we can do with ourselves. And this conjunction of bacteria are implicated in a lot of different pathologies like obesity, cancer, neurodegenerative disorders. And over the last decade, it has been increasing research in this field because it looked like it's associated with a lot of occurrence and also with increasing certain type of bacteria in different pathologies.
Okay. And can you maybe tell us, so we explained, we highlight some aspects of Preventomics, the genetics, the circulating food intake biomarkers, but in what way is the microbiome studied in the Preventomics project? What do you want to achieve with that?
Yeah, well, first of all, we had access to the fecal samples that have been collected during the intervention studies. And this give us the opportunity to characterize the microbiota composition, both at the start and also at the end of the intervention. And we're looking, first of all, for differences between geographical location, and after that we also looked at differences between dietary intervention, and yeah, we'll finally also looked at differences that have been differences between at the start of each intervention and the end of the intervention. We want to understand if the microbiome of the people that have participated in the dietary intervention have changed over the course of this intervention, in which way have changed also. And in addition, we performed also fecal microbiota transplantation in an animal model using these samples collected from the volunteers that have participated in the Preventomics project.
Yeah. And so, by performing these transplantation studies, is then the idea that you really would see whether the microbiome will influence maybe the direction of say weight development in these people, or are you interested to correlate the microbiome with what happens in circulation? Can you say something about that?
Well, our main objective with the animal model was to investigate if the microbiota of the post-intervention volunteers has the capacity to influence the phenotype, the microbiota, and the metabolome of an overweight animal model. And basically, we want to see if the changes in the microbiota after the dietary intervention can be translated to the animal model, and if this different microbiota can influence the outcome in the animal model.
Okay. Thank you very much, Radu, for introducing yourself in this nice way. I will now shift to Nuria Canela. She's actually head of the genomics, the Omics technology platforms, and maybe as a kind of introduction. So you analyze urine, and you also analyze, say, blood fractions. So how what type of metabolites are you looking for? Are you measuring?
Sorry. First, thank you to invite me, and it's a pleasure to be here. So in urine, we analyze forward metabolites, and in plasma, we have made different analysis. We are performed the analysis of the metabolome by NMR, nuclear magnetic resonance platforms, and for plasma, we have performed two different types of extractions. So for one side, we have extracted aqueous part of the plasma, and for the other side, we have analyzed the lipidic part of the lipidic part of the plasma. So we have different panels of analytes, and for the by NMR, we can get an overview of the major classes of lipids, not maybe the specific species, but we can obtain this overview of the lipid classes, and for polar metabolites, we can get different metabolites like several amino acids, glucose, and others compounds that are useful to get this whole picture of how it's changing the metabolome.
Yeah. Okay. Thank you for explaining this. And now the more difficult question. So you have seen the development of these technologies over the past maybe 20 or years or so, and so maybe you can say something about, say, the state of the the technologies at this moment? What has it particularly improved? And maybe you can also say predict how it will look in, say, 5 or 10 years.
I don't have the ball, but
let's try.
Yes. So, well, maybe the most developed omic technology nowadays are genomics and transcriptomics, so the genomic related technologies, and thanks to the improvements in next generation sequencing, that it's the larger scale DNA or RNA sequencing technology, which can characterize the entire genome or transcriptome. And next generation sequencing is now more cost effective and time efficient than previous techniques. So if we go to the 2000s, when the Human Genome Project is finished, maybe in 2003-4, it appears the first next generation sequencers, and the continuous improvement of this technology has dramatically changed the DNA sequencing industry and research. For example, at the beginning of this century, sequencing a single human genome took several years to be complete and cost more than $3 billion. And these days it costs maybe about few $1,000 or less, or euros, and with a few days of work. So this is the most advanced omics technologies now, but proteomics and metabolomics, it's also emerging as a very potential techniques. And in proteomics and related technologies based on mass spectrometry, this has revolutionized the field of proteomics, and now we can identify proteins, quantify, characterize their structures, and also or even analyze protein-protein interactions in a high throughput manner. But they need some improvements, and they need more. They need a lot of sample preparation, and it's highly cost. And finally, metabolomics, based even on mass spectrometry or nuclear magnetic resonance, has been also improved during the last 10 years with new instruments with higher sensitivity, higher accuracy, and more resolution. But in this case, the pipeline of data analysis for untargeted or global metabolomics studies are tedious and requires so much time to get a correct identification of the compounds. It's a time-consuming technology. So I hope in the next 10 years, the technology will continuously improving, and that will appear new bioinformatic tools to facilitate metabolites annotations. And this can facilitate the multi-omics profiling and to get more studies in personalized nutrition and personalized medicine.
Yeah, I know a lot of people are measuring metabolites, basically using almost the same technology, for example, NMR, and everyone is basically setting the standards to these peaks. Do you think that a kind of common database for that would be helpful, or?
Yes, we are working in common databases, and we are introducing every day new standards in the analysis to get a semi-targeted analysis to be easier to identify exactly the compounds. I think it could be a good way to work.
Okay, thank you very much, Nuria. I will now go to Josep Maria del Bas for a kind of follow-up question. So we've heard all the different type of methods and technologies and approaches. And my first part of the question would be, do you think they really can help to, they're really sufficient to describe the physiological state of someone? It's a difficult question. It's a large question, but maybe you can speculate on that.
Yes, well, it's really a difficult question. We have seen Professor Ordovás lecture, and how difficult is it to be able to characterize an individual, and the many aspects, the many dimensions that we have to keep in mind when doing this characterization. Actually, we have different ways of doing so. Professor Ordovás also talked about the concept of metabolic flexibility and how can we have information about the subject by challenging him or them with specific stressors and looking at the response of their metabolism and how the homeostasis is able to restore the influence of these stressors. And this kind of analysis would allow us to shorten the different, the timing that is required for all these studies and for characterizing the subject. And coming closer to Preventomics, well, what we realized was that with a combination of different kind of biomarkers, we could be able to evaluate these different processes that were selected for the project, with carbohydrate metabolism, lipid metabolism, inflammation, oxidative stress, and some biomarkers. Using some biomarkers of microbiota status, we could also have an idea of what could be going on with microbiota. So at this level, we can rely on clinical markers, but also on emerging markers, which are, let's say, predictive of the onset of different diseases. And this is the approach that we have taken in Preventomics, to have this combination of already used clinical biomarkers and some others with sufficient scientific basis to use them. We have relied mainly on meta-analysis or large reviews in order to select these biomarkers. So overall, I think that to have a complete view of what's happening with the individual at the physiological level, these tools are really, really powerful, and we can extract a lot of information from them. But we have to keep in mind other dimensions of the individual, such as dietary habits, for example, such as behavior, and in order to choose which approach is the best one for each one, right? So we need to integrate all this kind of information and all these kind of tools in order to have a wide view of what's going on with a specific person.
Okay, thank you, Josep. I think this is a very clear answer. I realized that I forgot to introduce myself. I'm Jaap Keijer, professor in human and animal physiology at Wageningen University, and my expertise is molecular physiology and metabolism. I would really like to ask everyone, there's the possibility to ask questions to these experts. Now you have a clear idea of their backgrounds. They all participated in this project, Preventomics. Maybe I think really made a big step towards, say, giving people advice on personalized nutrition. So if you would like to know anything, how their expertise actually contributed to that, don't hesitate to put any questions in the chat, and preferably to organizers and panelists, so that we can all see the questions. So don't hesitate to ask them. As there are, I will first do a few quick questions to all the different people. So I will go I will make the same round. Pedro, I will start with you, and maybe you can say something about the challenges that we are faced to measure dietary markers. And maybe also some things that the way you overcome this in this some parts in this project, or try to work on that. Maybe that's you could make a comment on that. Too big question again.
Thank you, Jaap. So we have to discuss about the things that at the end of the day complicate all the picture, no? I mean, first of all, I think that the idea at the beginning of the project was quite ambitious. And actually, I think Josep commented on that. There are many biomarkers, both in the health side, that in the food biomarker of intake side, that has not been validated. No, and our first limitation was to set a realistic panel, let's say a reliable panel of biomarker of food intake, because today there are just two, for me, validated biomarkers of food intake. But we wanted to, one is for apple and the other one is for citrus, but we wanted to profile the whole diet of the population, no? So because of that, we have to rely on some biomarkers that are not robust enough. But the importance of this project is that we will be able to, using all the intervention studies, to try to validate this biomarker. So it's a novel field. That means that you have to do a lot of things by yourselves. But at the same time, we're developing let's say, a rocket science using obviously the contribution of previous researchers. So the lack of validated biomarkers was a limitation. Then the analytical constraints of these biomarkers. You want to analyze many different compounds, as Nuria said before. Not all the compounds can be analyzed in the same condition. So because of that, you also have to try to set, let's say, analytical platform able to cover all the metabolites you want to cover, having the quality in the analysis that you need to proper address the task that in this case is profiling the diet of a person. And then I would say that another point, one of the limitations is related to the fact that at the end of the day, this biomarker, and that's an intrinsic limitation of this tool, is that they provide you an overview of the compounds you have in circulation, probably related to the foods that you have been eating in the last day or the last 3 days. Most of the foods at the end of the day are cleaved are released from your body. Let's say the metabolites or the compounds present in the foods, once you intake them, they consume them, they are released from your body after 1-3 days. So we cannot rely on what a person has been eating in the last 15 days somehow. But as I said before, in previous intervention, by having different samples, we are able to provide a good overview of the diet of the population. So that kind of limitation in terms of analytics, in terms of the nutrikinetics of the compounds, let's say the travel of the nutrients in our body, at the end of the day, that's something that we can cover by using proper approach in terms of sampling. And I think that the good point of this Preventomics project is that we have been able to improve very much our capabilities. I think that we will improve pretty much the capabilities of the whole field when we publish the final result, that to date are very encouraging. And I think that, I mean, we discussed pretty much about all the limitations. We suffer pretty much. Jaap, now or today, all we are happy, but we have been discussing pretty much about the quality of the analysis we are doing and so on. So at the end of the day, today it's a party, and despite of the limitation we face in terms of practical arrangement, and then also in terms of scientific constraints, I think we have reached an incredible level.
Yeah. I think it's I'm very pleased to hear that you say, so basically we started with nothing, and actually that this project really will help us to build a kind of comprehensive overview. You also mentioned the limitation of the short terms. Do you think that also measuring not only in blood and urine, but maybe possibly in white blood cells, that will extend the time where you can have an overview of the dietary exposure?
I mean, I think that the more biological fluids or sample you have, the better. Not even physics can be very interesting, because at the end of the day, they can provide also a good overview. When it comes to white blood cells, for some compounds, they can be key. I'm sure we don't have data on that, and that could be the next step, not try to better understanding which biological fluids we can have a better performance of our biomarker of intake. But of course, the more sample you have, the better. And obviously, you have to deal with the characteristic of some compounds. Some compounds that are not absorbed will be better characterized through physics, and some compounds that perhaps remain for a lot of time in circulation because they are binding to white cells can be, of course, of interest. But I think that's a matter of discussion for the future.
I have now a question that both you and maybe Radu can answer. So you also mentioned the features that you can measure metabolites in there. That will also change the microbiome. Radu is really looking at how the microbiome influences the physiology. But do you think you could also use the microbiome as a kind of marker of of nutritional exposure, or is it more related to the individual physiology? It's not an easy question, so one of you may answer this.
Okay, can I answer, Pedro? It's okay for you?
Absolutely, Radu, it's a question for you.
Okay, thank you. Well, Jaap, probably I would say that this is the biggest question in the field of microbiome right now, because the underlying mechanism of gut microbiota and human diseases are incredibly complicated, and it really depends on which affection or which pathology we're talking about. But for example, for obesity, overweight, and overweight, which is more related with this project, what is really clear is the fact that the microbiota composition between lean and obese individuals is different. But how this trying to answer if certain type of bacteria predispose you to an overweight phenotype, let's say because you have, I don't know, maybe initially more actinobacteria or more proteobacteria, or you gain more weight because of that, or is the weight gain which leads you to increase this type of bacteria? It's a very complicated question, and we really don't have a clear answer yet. But what it's really clear is the microbiome can be modulated mainly by the diet, which is the major key role in the microbiota composition. And for this reason, in this case, the diet intervention are a great way to change or to reduce certain type of bacteria that are associated with pathologic phenotypes like obesity in this case.
Okay. No, I think it would be great if you would see how these exposure markers also correlate, and then also with physiology, and maybe it's possible to disentangle the things. I would like to go to Francisca, and so Francisca, you said that in the beginning you started with a few markers. Now you have several hundred genetics markers. So can you maybe explain how you made the selection to go from a few to a larger number, and then so what your strategy is, and maybe then also if you have the different genetics, what is your strategy of translating this to people actually?
Yes. The strategy has been, first of all, to look at very big studies, what is called genetic-wide association studies, that may give a very strong correlation about the relevance of the genetic variants that we selected. And in these studies, what is shown is the relationship between the presence of the genetic variant and a phenotype. And for example, a phenotype trait, as can be high levels of cholesterol, or can be tendency to obesity, or tendency to when you follow a diet, the tendency to slim in a worse or in a better way. So we have focused on these genetic variants that could be found in the scientific literature. And then we have selected or make three big groups of variants. One is the group of genetic variants that can predispose to a specific tendency, for example, to obesity, to diabetes type 2, or to dyslipidemia, and so on, the parameters related to the Preventomics project. The other set of variants was devoted to know which is the best nutrient or diet for your genetics. So was based on the interaction between genes and nutrients. So if we have some diets that can be efficient to lose weight, which is the best for you? So we can make an estimation concerning the genetic background of the person and how this person would respond to these different interventions. And in this way, we give a prioritization. So this is the diet that could be the best for you, and this in the second place, and so on. So with all the opportunities. And another set of variants are have been analyzed, but haven't been included in the report to the patients, because they are very interesting variants in scientific literature, but we think that we don't have enough evidence to give an advice to the individual persons. So the genetic variants have been studied, and we think that in the near future we can confirm that they are relevant, and then we could add this information to the patients, or to the nutritionist or the health professional. So I think this has been the setting up of our platform. Concerning the technology, we didn't have the problem that Pedro commented on the biomarkers, on dietary biomarkers, because the technology to determine genetic variants is already well documented, and has been setting up for the last 20, 30 years. So in this case, we already were sure about what they need to use. And our work was to select the variants, because there are millions of variants that you can study. So we have to focus on those that could be important, and on those that we could have an interaction that could give a person a positive advice. You have these variants, but on this way you can modulate in a better way for your benefit.
Yeah.
I think that I don't know
Yeah,
if I
so
respond
yeah.
to you
So
the
there's
question.
actually a question
More or
that
less
is
that
a
was
question in the
our
chat,
setting up.
and it is from Alexei Levici, and I think it's a very interesting question, and it may be not only it's directed at you, Francisca, but it maybe applies to all of them. So the study for the genetic variants, did you focus on, say, a Caucasian population, or did you take different ethnicities in the background, and to what extent, maybe extend it also to other people if they would like to comment, is this taken into account in the Preventomics project? I think it's a very nice question.
I can start. On the
Yes.
genetic variant, the selection of genetic variants was based on the scientific reference, and most of them are based on Caucasian people, but also there are a lot of information on Asian people. In the estimation of the tendency, for example, to obesity or to dyslipidemia, we made a normalization process. And in this process, we used the data on European population that are around 500 genetic biomarkers published from this from person of this characteristic. But we are aware that if the results of this project have to be delivered to other communities, we should normalize for the genetics of the specific populations. This is a very important project. And already in Alimentómica, we have started to develop this aspect. So we analyze the genetic background according to the ethnic of origin of the person. This is an important issue.
Okay. So the ethnicity, there's another question. I will come to that, but I just have a more a general question. So we know that in this type of studies, actually the interindividual variation is really big, and I think when we go to a disease state, it's maybe more easy, because people really have a disruption of their physiology. You have really big changes, and maybe then that overrules the inter-individual variation. But here we are in this in a project like this, we look actually at the pre-disease state. So we have people that may become obese or are slightly obese, but may also be healthy and wanting to lose weight, for example, and just to go to a more healthy state. So what is the, do we have enough sensitivity, or what are approaches to actually disentangle this large inter-individual variation? And maybe Josep, you can first comment on this. Also, what maybe strategies in the project, and then others may add to this.
Yes, well, what we have done is to add different layers of personalization in order to have all these different domains of dietary habits studied, and also physiology state. So we have used genetic information, as Francisca has explained, in order to know these differences between subjects. We all know this. Well, there are famous experiments with twins with the same genetic background, but different habits and lifestyles, which one of them develops obesity and the other one remains normal weight. So we have had this genetic background into account when designing all the different interventions. But also we have gone further to study what's the context of the subject in terms of dietary habits by means of questionnaires and so on. We have keep in mind as well, we have considered as well the behavior of the person by means of behavioral change programs, and also adding more and more layers of personalization, such as adherence to the Mediterranean diet, which was considered the healthy diet in this project, and of course the metabolic state. So with all this information and integrating all this information, we try to differentiate between these interindividual variations.
Okay. Does anyone else would like to comment? Otherwise, I would like to use the first minutes from a question of the audience. And that is really the question: can we match a specific diet to the microbiome to get a better weight management? I think this is what we all would like to achieve, but maybe Radu, you can comment on that, how achievable that would be, or maybe in which time frame.
Well, it's a very nice question, very interesting, but at the end of the day, we don't really have a gold standard diet in order to shift the microbiota composition to a healthier state, let's say. There are described a lot of new ingredients and a lot of new food consumptions that are associated with certain type of bacterial genera that maybe may enhance the production of shorty chain fatty acid, or maybe are more like probiotic bacteria, and this type of compounds can really help to enhance this type of beneficial bacteria. But at the end of the day, it's not. We don't really have yet a golden standard. What really impacts the microbiota to have a healthier way, it's of course what we eat, and the vegetables, what are the behavioral changes, what we are taking into account, sport, don't have a lot of stress and things like that. I would say it's a combination of both, of a lot of factors that can really help to have a healthier microbiome. We don't have yet a magic pill to say take this and you will have great microbiome. And, okay. And for this, it's much more research is needed in this field.
So As the time on the schedule regretfully is over, we had still quite a number of questions also for the audience. If there are additional questions, just type them in the chat and we will get back to you by email. I didn't think I gave everyone as much time, but it's really quick. I would like to thank the audience for their presence. So what we have heard is that in the Preventomics projects, we try to integrate basically exposure, genetics, circulating biomarkers from blood and serum, and also markers from urine to recover the metabolism. We integrate the microbiome and we use, say, informatics tools to get the best data out of that. And it is in the project, this is all basically validated in 4 human studies to see if we implement these the data that we get out of this, how this will basically result in a outcome. After the break, which is now until 10:30, you can hear more about both these human intervention studies and the results out of that, and how this project was, how the data from this project were used. And on the other hand, we will also explain more at all the digital tools that we developed. And this ranges both from integrating this data, but also how we communicate with the consumers and how they are actually in charge of their own dietary choices. And I think both of these sessions are highly interesting. I recommend them, but regretfully you have to choose. But that is like with a lot of life. Finally, I would like to thank all the panelists for answering the questions and being here and share your experiences. And of course, the audience, thank you very much for adhering to this lively session. Okay, thank you. Bye-bye.